Good afternoon, everyone. Thank you for joining our webinar this afternoon. This is a monthly webinar series that we do from the Duke Cancer Institute Office of Health Equity. And this is the series we call Conversations with Community. So this is one way that we bring some of the expert knowledge of scientists and researchers from Duke University and allow them to be able to share some of their expertise with the community. And at the same time, we want the community to share some of the questions that they may have as you interact with the different presenters that we have scheduled for us. Today, we're going to focus on the title of this session is How Studying Genes Can Lead to More Personalized Cancer Care. It's really important that we really understand genes. There's been a lot of conversation about how important genes are and how we take care of patients, specifically cancer care. So I've seen the presentation. I am sure you're really going to enjoy and learn a lot from this presentation. So this is the agenda we're going to follow today. I'm going to introduce the speakers, take about 5 minutes. We're going to have the presentation and each speaker is going to present. We have 3 speakers today. After that, we'll have a question and answer period. But I encourage you throughout this presentation that you put your questions in the chat. So we have people that's on the line. Hopefully we'll be able to answer your questions. If there's questions, we don't have time for questions at the Q&A session, I'll get with the presenters and they can give me answers and we can send those answers back to you, their responses. We're going to spend about 5 minutes just to close out and thank you all for attending. So our first presenter is Dr. Jennifer Friedman. She's assistant professor at the School of Medicine at Duke Cancer Institute. Dr. Friedman received a Bachelor of Science in Biology and a PhD in Genetics and Molecular Biology from Emory University. She also received a postdoctoral fellowship at Duke University School of Medicine in Cancer Biology. She co-directs a translational research laboratory with Drs. Paterno and Dr. George, which focuses on the preclinical studies and the correlative science to help explain the molecular mechanisms underlying cancer disparities and develop novel biomarkers and the therapeutic agents based in these mechanisms. The laboratory's ultimate goal is to develop novel approaches for prevention and treatment that will reduce cancer disparities and improve outcomes for patients of all races and ethnicities with aggressive cancer. The second presenter is Dr. Tyler Allen. He's a postdoctoral research fellow and associate in Paterno, Friedman, and George lab at the Duke Cancer Institute. Dr. Allen, he earned his bachelor's degree in biology from North Carolina State University and a PhD in biomedicine from the College of Veterinary Medicine at the North Carolina State University, where he was part of a team that discovered a novel mechanism, certain cells employ to exit blood vessels, termed angiopoiesis. His research project focused on the discovery of how circulating tumor cells utilize this method to spread cancer in patients, what we call metastasize, leading to what is known now as the cancer exodus hypothesis. In addition to being a scientist, he devotes time to writing, consulting, speaking, and STEM outreach with the focus on inspiring and engaging the next generation of leaders and change makers. The third speaker is Dr. Sean Prworski. He's a postdoctoral research fellow and associate as well in the Paterno Friedman George lab at the Duke Cancer Institute. Dr. Prworski completed his graduate studies at Marshall University, earned a PhD in biomedical sciences. His research project focused on identifying novel cancer therapies by understanding how environmental toxins and compounds regulate cell signaling pathways in breast cancer. At Duke, his project involves developing a dual fluorescent mini-gene reporter system to study the alternative splicing of specific exons in African-American and prostate cancer, as well as differentiating and analyzing metabolic profiles of African-Americans and Caucasian prostate cancer patients. In his free time, he likes to spend time with his fiance and 3 dogs, as well as working part-time as a freelance artist. So as you see, their whole life is not all science. They are human beings that do other things as well. So we really look forward to their presentation today. And at this time, I will turn it over to Dr. Freeman.
Thank you so much, Dr. Moore, for that kind introduction. Can you all, I just want to make sure everybody can see slides. We're good. I see Dr. Moore nodding. Okay, fabulous. Thank you so much for that kind introduction. And thanks so much, everybody, for joining this evening. Most appreciate you joining, especially virtually. It's great to be able to get together. We really appreciate the invitation. We're super excited to be here and for the opportunity to share our work, some of our work today. So thanks so much. And as Dr. Moore said, we really want this to be interactive, and we really treasure any input we can get and really welcome any input we can get from community members and from the community. We really want to work with the community side by side to address the questions we're trying to address and do all the work that we do. So please feel free to type any questions in the chat and ask us any questions at the end. And also, provide any input as well that you might have that we would definitely welcome being able to use in our work. So as Dr. Moore mentioned, we're going to talk today about the work that we're doing to study genes and how this information can lead to more personalized cancer care. And this is super important and, really important, as I'm sure you all know, to personalize cancer care, in large part because we have a lot of evidence in cancer and also in other diseases that it really needs to be personalized, that members of different population groups get cancer at very different rates. They have cancers that vary in aggressiveness, and they also respond to treatment very differently. And so we really need to personalize cancer care as much as we can. And this goes for other diseases too. I think, this topic is very, very timely because I think, as many of you I'm sure have heard, that the COVID-19 pandemic has really brought much needed attention to this challenge that we have, that a lot of members of population groups get diseases at different rates. Like I said, they're more or less aggressive and respond differently to treatment. And this has been true in the COVID-19 pandemic, where we've seen that members of the Native American community, the African American community, and the Latinx community, for example, have suffered disproportionately from COVID-19. So similar to the kinds of things we're thinking about, that's true with cancer as well. So just see if I can advance slides. Let me share again, see if I can, sorry everybody, see if I can share my screen again and get slides to advance. Okay, can everybody see the second slide? Okay, great. Sorry about that. Technical difficulties. Okay, so like I said, we're focusing in cancer, and definitely in cancer, we see evidence of these disparities, that members of certain population groups suffer disproportionately from certain cancers. And shown on this slide is just some examples that we'll run through of examples of cancer disparities, just to give everyone a feel for this. So for example, in breast cancer, African American women are nearly twice as likely to be diagnosed with triple negative breast cancer, which was one of the more aggressive and worse forms of breast cancer. And they're more likely than white women to die from breast cancer. Kidney cancer, we see a similar trend among African Americans, and also the highest rates of kidney cancer cases and death in the United States occur among American Indians and Alaska Natives. Same with liver cancer. We see those rates being higher among American Indians and Alaska Natives, as well as Asian and Pacific Islanders. Prostate cancer, African American men are more than twice as likely as white men to die from prostate cancer. They are also much more likely to get prostate cancer than white men. We also see these disparities in urban versus rural populations in certain cancer settings. So for example, women in rural areas are twice as likely to die from cervical cancer as women in more urban areas. And again, in multiple myeloma, African Americans are twice as likely as whites to be diagnosed with and die from multiple myeloma. And I just want to mention, in terms of racial and ethnic disparities, I pulled this slide from cancer.gov website, a national website on cancer through the National Institute of Health. But I do want to mention that cancer disparities don't always go in the direction of members of minority groups getting cancer more frequently or dying more frequently. And an example of that, a really good example of that, is in brain cancer or glioblastoma, where whites are much likely to get it and to die from it than members of other racial and ethnic groups. So it can really go in multiple directions, as well as urban. And so these disparities influence patients' risk of getting cancer, how aggressive the cancer is, how bad the cancer is, and it can also influence, like I said, response to treatment. So there are many factors that influence a person's risk of getting cancer, how aggressive the cancer is, or how that cancer responds to treatment. And so I just want to highlight examples of these factors. So the first factor is based on our individual level genetics, or our genes, which is the predominant area we're going to focus on today, as the title of the talk reflects. And these can also be, very importantly, can be ancestry related. So, throughout history, as people migrated, and this could be through voluntary migration or through forced migration throughout the world, our ancestry changed. That's why, members of different populations, definitely we look different. And so that the different looks that we have on the outside also reflect differences on the inside and in our genetics and our genes that are ancestry related. And so this causes us to have very, a lot of differences in terms of the physiology in our bodies and our genetics or our genes or our DNA. And so, like I said, we're going to focus on those differences in the talk today. But I do want to mention other factors that we know also contribute to these disparities. So other factors are society level factors, such as racism and discrimination. In addition, we have neighborhood level factors such as diet, lifestyle, the environment, pollution, and we also have institutional level factors such as the access, for example, the access that people have to getting care. So all of these factors really are genetics, what's happening at the society level, what's happening in our neighborhoods, and what's happening with our access to institutions, all influence our risk of getting cancer, how bad our cancer is, how it progresses, and how we respond if we have cancer, how we respond to treatment. So like I said, we appreciate that all of these factors contribute. We're going to focus today on individual level ancestry related factors, or our genes or our DNA, but with the constant appreciation, and we'll come back to this at the end, of knowing that we really need to think about all these factors together when we're doing the work that we're doing and how they interact. So I just want to mention briefly that cancer disparities occur throughout the cancer continuum, as we call it. So we can see evidence of cancer disparities with respect to prevention, early detection, how we diagnose cancer in people, how people receive treatment and their response to treatment. They extend out past cancer into how people deal with survivorship, and also even into end-of-life care if it comes to that in cancer care. So we really think of disparities across this whole area, although we'll be focusing today on disparities at the level of cancer diagnosis and treatment. So there are racial and ethnic disparities, not only in cancer, but also in cancer research that really need to be addressed. So there have been 2 recent studies that have really highlighted this. And one has shown that in projects that we do in laboratories that focus on genes, which, and cancer, which is the topic of our talk today, a lot of the studies that we do rely on samples from patients. And what we see is that in these studies, the samples that we have in these that are currently available to us are overwhelmingly from white patients and really underrepresent patients of other minority groups. And so we really need to work on this and get more samples from patients that reflect the diversity in society so that we can really make sure that we're studying disparities and we're able to address disparities in cancer. And the same is true on the clinical research level. So if we look at clinical trials and specifically clinical trials that have led recently to cancer drug approvals, we again see that the participants in these trials are overwhelmingly white and underrepresent patients of other minority groups. And so we really want to do this better as well so that we can make sure that we're, again, studying disparities correctly and addressing disparities. So the work that you're going to hear about today is work that we're doing to really identify genes in cells that drive cancer disparities, which we really can think of as the targets, and then develop treatments that block these genes so that really are the dart that can hit these targets. And so we want to use information about genes in an individual's cancer to develop drugs for cancer. And so here we just see a cartoon of a tumor where we have normal cells, and then within the normal cells we have a tumor of rapidly growing and dividing cells. And eventually some of these cancer cells can detach and spread to other parts of the body. You may have heard of people who have had cancer start in one place and spread to another place, this is what we call cancer metastasis. And so we can, to study how genes are influencing cancer, we can compare genes in patients of different races and ethnicities. We can look in normal cells versus cancer cells at genes. And we can look at less aggressive versus more aggressive cancer cells. So cancer cells that grow or spread less versus grow or spread more, and look at differences in the genes in all of these cases. So I'm going to use an analogy to talk about some of the challenges we have in studying cancer disparities of a cheeseburger. And I apologize to anybody who has not had dinner yet. It was probably a very poor choice of analogy at this time of the day. So hopefully it won't make you too hungry. But we can think of cancer disparities and the challenges in the context of a cheeseburger. And we really need more fixins. We really need more darts and more targets. We need more of an understanding of genes that are important for cancer disparities, and we need more drugs that we can use to target these genes. Just like people, cancer gets tired of the same fixins. Cancer drugs stop working. You may have known people who are cancer patients where drugs initially work well, and then they stop working, or in our field, we call that developing resistance. And so we need to get more darts and targets so we can deal with resistance. And then also, just like people like different fixins on their burgers, different cancer patients need different combinations of fixins. So again, we need to do combinations of personalized cancer care where we have the right targets and the right darts in each person. And so this is just an overview of our research program. We use tissue and blood from patients of different racial and ethnic groups. And then we use these samples to get at genes that are driving cancer. And Dr. Allen and Dr. Pawlowski are going to talk more about this, all of these aspects in just a minute. And we develop tools or drugs to manipulate genes for cancer treatment. We are developing cancer models that we can use in the lab from patients of different racial and ethnic groups to study the cancer more specifically. And we're also conducting race and ethnicity stratified clinical trials to get at the role of all of these genes and drugs, looking at it in clinical trials. And we're going to focus today on some examples of our work in prostate cancer, but we're doing a lot of work in lung cancer, as well as liver cancer and breast cancer. So we're really, a lot of the same concepts that you'll hear about today in prostate cancer are also being done in our lab in the context of other cancers, like those that I just named. So with that, I'm going to turn it over to Dr. Allen to talk a bit about his work.
Thank you, Dr. Freeman. I appreciate the turnover. So real quick, so I'm going to talk about kind of the genetic, or kind of give a brief overview of the genetic component, or the genetic side of what our research is focusing on. So before I start, I just kind of want to give you all a brief overview of just kind of the central dogma, as it's known in biology, which talks about so, you know, each of us, we're humans, we're a person, but inside of us, we all have DNA. Click to the next slide. So we all have DNA in our bodies, and each of, you know, of our cells has DNA. And so as you all might remember from like high school biology, you know, the DNA is the piece, or pretty much the piece of, I guess, thing in our body that tells our cells in our body what to do. And the DNA makes RNA, and then the RNA makes the proteins, and then the proteins are pretty much what do all these functions, all these different functions in the body. And so we study kind of how these genes at the DNA level are affecting these processes in terms of how it's making RNA, and then how these RNA are making proteins, and then how these proteins are impacting or affecting certain diseases, in our specific case, cancer, and then also how they're affecting patients' response to treatment or to cancer. So moving on to the next slide. So our lab specifically focuses on a process known as RNA splicing. So thinking back to that diagram, remember I said the DNA makes the RNA, and then the RNA makes the protein. So that's the kind of the simplified version, but there's a little bit more that goes into the process. So once the DNA makes the RNA can actually make, or can be put together in certain ways to create certain or different types of proteins from the same piece of RNA. So let's move into the next paragraph. So the way that the body can make proteins from RNA can change depending on how the RNA is put together. And this can change between how, or different people or different populations, specifically like racial or ethnic groups, can use or can put RNA together differently to make different proteins, which can then have different effects in the body. And so that's the essence of what our lab is working to study now, is how RNA is put together differently in different ethnic or racial groups, and how this is affecting the types of proteins that are being created, and then how this in turn is affecting the rates or the types of cancer, or the way that the cancer is progressing in different patients of ethnic groups. And so there's been a lot of recent studies that have shown that RNA splicing does, or I was gonna actually, no, I wasn't finished with that slide. I was gonna say just real quick, so you can think of RNA splicing as like a Lego building block. So we all have DNA, all of that DNA gets made into RNA. And so the RNA is like a bunch of building blocks. So even if we have the same Legos or the same pieces of RNA, we can put or arrange those DNA or those RNA pieces or those Legos in different ways to make different objects that have different capabilities. So that's a good way to think about RNA splicing, is the same components, but just put together in a different way and have a different function. So now moving on to the next step. So why is this important to study, or why are we even interested in this? And one, kind of like I mentioned, is that there have been multiple studies that have come out in the past really few decades that have shown that between different racial and ethnic groups, the splicing, or the way that these Lego blocks are put together, is different. And additionally, not only is it different, but these differences are causing variation between how diseases progress and then how these diseases are actually being able to be treated by therapies and drugs. So I guess next, or moving on. So, and yeah, keep, or one more. And then, so pretty much, I guess the importance of this type of study is showing if we can better understand how these differences in RNA splicing are happening, we can better understand why potentially we're seeing such disparities in racial or ethnic groups in the context of certain diseases like cancer and even other diseases, because RNA splicing can affect a multitude of different processes or things or diseases in the body. So this is a really important and kind of more new field than some other fields in biology, but there's a lot of promise to hopefully addressing some of these disparities that we're seeing beyond just the other levels that Jenny had mentioned before, and kind of looking at what genetically could be accounting for causing some of these differences that we're seeing in disparities in the role. So one kind of good example of this is cancer metastasis, which Jenny had alluded to previously. And so that's just the spread of cancer. So for example, if someone has a cancer that originates in their lung, sometimes that cancer can actually spread throughout the body and then can potentially start forming new tumors, maybe in the liver or even in the brain or other areas in the body. And so this is it's a highly regulated process in the body, but if we could advance one slide, and then another. But this process has been shown to be impacted or regulated by RNA splicing. And kind of some of the projects that we're working on now in the lab is looking at how RNA splicing or how this process is influencing the spread of cancer, and how this is potentially different in different racial or ethnic groups. And so just an example of one of the reasons why this is such an important type of area of cancer to study RNA splicing, because, for example, if we were able to identify how RNA splicing differs between patients, we can develop therapies or drugs that are able to better treat or target these cells or these cancer or tumor cells, preventing them hopefully from spreading, or at least treating them after they've already formed in the body. And then also looking at how this is different between racial or ethnic groups, and if there are any differences that we can pinpoint or target to help kind of decrease or address some of these cancer disparities. And then moving on to the next slide. So, okay, so I've given you the what and the why we're studying this, but now we're gonna, I guess I want to talk about how we actually study this in the lab. So say we have, I guess, some splicing or some RNA splicing studies that we want to look at. So what we can do in the lab, or what we actually are doing in the lab now, is a lot of the projects or research that we are working on start with identifying patients who have either had cancer or have cancer, and we are able to get or collect samples or tissues from these patients. And these are actual like patients that may live in the community or even internationally, and we're actually able to get samples, either tissue or cancer or tumor samples, or sometimes just normal other blood samples. And then we're able to take these into the lab and do analysis on these samples. And what we can do is we can look at the differences between these tissues and see if we can identify any genes or any parts of the DNA that are different, or expressing or showing differences at the DNA and RNA level. And so then once we have that, we can take cells, or we can pretty much artificially change cells in the laboratory to mimic the changes that we're seeing in these different types of tissues. And then we can study in the lab how these different genetic changes are affecting how the tumors or how the cancers behave. just in general. So the hope is that if we understand how they're behaving, we can develop, or we can start to develop therapies or drugs that can target and actually prevent these cancers, or treat these cancers once they've formed, or once they've started to progress in cancer patients. And so one of the, I guess, ways that we can manipulate cells in the lab is through a process known as CRISPR. So this is actually a very interesting and cool process. And actually, the founders, or the kind of discoverers of this process, recently won a Nobel Prize for their work on this. And what it enables us scientists to do is we're able to take cells in the lab. So this is a cartoon illustration of a cell. We're pretty much able to open up and look at the DNA, or actually open up and modify the DNA. So if we have a gene that we're interested, let's say from the patient samples that we've acquired, that we've gotten, we identify a certain gene seems to be different in certain populations. We can go into the DNA and then modify that gene in cells in the lab, or cells that we grow in the laboratory. And we can change that gene to mimic what we're seeing in the actual cancer patients, and then see how that affects the tumors or the cancer cells in the lab. And then that can actually inform us how potentially these tumor cells or these cancer cells are behaving in patients, or patients that have, I guess, these same or similar types of things in their DNA that we're seeing. So this allows us to manipulate cells and kind of test them in the laboratory, and then go back and hopefully develop therapies that can be used in actual patients in like the hospitals, or for treatments, or for clinical trials. So then, so once we have identified and we've studied these genes, or this DNA and RNA in the lab, we can then, with the information that we've gained from these studies, we can develop specific RNA or specific drugs or therapies that can target these genes that we've identified. And then these genes can then be, or these drugs can be hopefully reintroduced into patients in either clinical settings, or the hospital, or for treatments for patients who have cancer, or these type of diseases that can be studied with this. And then the hopes is that with this information, we can make informed decisions about what types of specific therapies can be used. And so in the lab, we've got a few different things that we're looking at. One are just some types of molecules that we can use to impact the way that these genes are being expressed in patients as a potential therapeutic for treating cancer patients, and then some other methods. But the overall overarching goal of this is to be able to take patients' information, patient samples, study these in the lab, and then use what we're finding in the lab to make informed decisions about what drugs or what therapies can we then reintroduce into the clinical setting, and specifically how what we're seeing in the lab is different between racial or ethnic populations, and how we can develop targeted or specific drugs to help address these specific disparities, or these specific differences between racial and ethnic groups at the genetic level that we're seeing, so that hopefully we can address some of the genetic components of this, I guess, cancer progression, or differences in cancer progression and disease progression that we're seeing. So with that, I will turn it over to Dr. Pawlowski.
Yeah, thank you so much, Dr. Allen. And I'm sorry, thank you so much for putting up with my slide advancing skills, challenging with one slide deck and multiple speakers. So thanks so much, Dr. Allen. Okay, great. Dr. Pawlowski is now going to talk a little bit about his work as well.
Okay, so a lot of my research actually obviously overlaps with what Tyler presented. So a lot of this is actually going to be easy to take in and reiterate. So I have a couple figures for you here. If you see the top graph here, the black line, we are looking at incidence incidence rates by race and ethnicity, the year of which people are races are diagnosed, and this per 100,000. And as you can see, the Black population has a much higher incidence of prognosis and diagnosis in prostate cancer. And if you look at the bottom chart, you could also see that along with the prognostic rates, you also see the death rates also are extremely high compared to other races. So over the years, there have been a conclusion that we can see that cancer disparities in different races, and especially in African Americans, the African American population tends to have a much more aggressive cancer they're diagnosed at a younger age. They do have a lower survival rate. They are also more likely to come back after enduring treatment, and as well as most likely to spread to other organs. So a lot of this shows that African Americans tend to have a tougher time when diagnosed with prostate cancer. But however, what's interesting about this is that African Americans, even though they have more aggressive prostate cancer compared to the white cohort, when they undergo the same treatment, African Americans tend to respond better. So even though it is more aggressive, the manner of how they respond is actually much more palatable compared to how the white cohort actually responds to cancer treatment. How come? Why is that? And that is the purpose of the Abbey Race study, where we actually collected 50 African American patients and 50 white patients that had metastatic prostate cancer, very aggressive. And we gave them the same exact treatment of abiraterone and prednisone, which is a secondary treatment to target the ability for prostate cancer to make testosterone and grow. So once they undergo the same exact treatment, we then take their serum samples, before and after treatment, and we actually run a biological analysis on those samples. So you can see the whole blood. We actually did genotyping, where we actually looked at DNA analysis. We compared the DNA profiles between the African American and white cohort, as well as metabolite analysis. So metabolite analysis comes from the diet, the way you break down drugs, the way you break down cells and everything. And that is also different too, depending on the DNA analysis. So with all the information that we gained from the biological aspects, that's when we go to the laboratory, and then we kind of put this, connect the dots and bring everything together and conduct our hypotheses in cancer cell experiments. So one thing we're going to go over is single nucleotide polymorphisms, also known as SNPs. It is very similar to Tyler splicing in the sense of the only difference is rather than you take out an entire Lego, you actually just change one of the little tiny nodes on the Lego. So instead of taking out a big chunk of DNA, you're only changing one base pair. So as you can see here, at the A and T group as the normal DNA molecule, it's perfect. You have this protein that makes, but then if you have a SNP where you just change that base pair, then you can actually see you still develop the protein, but the function of that protein may be altered. We know it may work better, it may work worse, but these proteins in a way may have a role in playing a role in metabolism, which is where you are what you eat. You take in molecules from your diet, whether they're from vegetables, food, fast food, all these proteins that actually have SNPs in it actually can regulate the metabolism of what you eat differently. So depending on these SNPs, you could also have a different outcome between races in terms of what you eat. And these particular metabolites also play a role in potentially inhibiting cancer or progressing cancer. And so when we did our SNP analysis, there's one particular gene that we found that is highlighted in yellow. There are 5 different SNPs within this one gene that is different between the African American and the white cohort. And it's involved in, well, what do you know? Metabolism. So from there, we actually moved forward, and we actually wanted to look at the 2 different metabolic profiles between the African Americans before they were treated and after they were treated, and then compare it between the races. So we have those, and we have the ancestry here. And what I do is that I take all these changes in the metabolites, and there's well over 700, 800 metabolites, I want to say. But the analysis picked up well over 500 metabolites. And so what we want to do is we want to put those metabolites side by side, and we run them into this software analysis program that looks at these metabolites, and then they actually go inside the deep dive of literature and science, and they say, oh, these metabolites are related to particular diseases and functions that promote cancer. Or, we can identify outcomes in terms of toxicity, as well as biomarkers. And as we see, when we take this bird's eye view of both the white cohort and the African-American cohort and put them side by side, we can actually determine how these biofunctions are altered. So as you see this chart here, when I did my metabolic analysis, the blue indicates that there is an inhibition, while the orange indicates there is a promotion. So in terms of cancer, you want cell cycle progression, which is, the cell being able to develop the necessities to actually proliferate, as well as cell proliferation in general, which is the growth of tumor cell lines. And you can see those are the two top cancer functions that are shown to be inhibited in African Americans as opposed to white patients. So that actually also lays claim to all the evidence that we've seen prior that African Americans tend to respond to treatment much more effectively than the white cohort. So with all this information that we gather with the SNPs and with this metabolism, so CRISPR, for instance, as that Tyler mentioned, one of our experiments is to actually take those different SNPs of our particular gene that we're interested in, that had those 5 different mutations, and we're going to CRISPR those mutations into these prostate cancer cells as a means to mimic the African-American population in a means. And then we're going to treat those with abiraterone, and we're going to compare that to the wild type, which is, the normal prostate cancer that, the white cohort has. So all of this is done in cell lines, and we try to connect the dots at that point. And we're going to try to see if there are different kinds of metabolites that they're responsible for that actually act as potential biomarkers, and as well as, identifying different types of experiments. I think, so yeah, so that's what we kind of gain from the information collected from this Abbey-Ray study, is with knowing how, the African-American population responds to treatment, could this lead to a new race-related treatment strategy towards precision medicine? What kind of biomarkers are present in African Americans that are metabolites? There are certain metabolites that have been identified to be pro-cancer, but then some other people just say, oh, I don't think it's pro-cancer. But at the same time, if you define it as a race relation rather than an overall population, that's how you kind of add credibility and specificity to precision medicine in order to develop novel treatment strategies for particular races, as well as identify potential biomarkers that are race related. Certain metabolites may be higher in other races compared to others that indicate prostate cancer may become aggressive. So knowing how we identify these metabolic profiles as well as these SNPs, we could put the pieces together and identify what we need to do and move forward from that point.
Great. Thanks so much, Dr. Perarsky, and thanks so much to you too for your patience with my slide advancing skills. So thank you bunches to you both. So just to wrap up, and then we'll have time for questions. And input for sure. So this is just the overall vision of our research program. We see these challenges and disparities that are in the community and the clinic, and we want to take that as motivation for what we want to study, and we want to study those challenges and those disparities by using information from the community and information we can bring into the lab. That can be in the form of surveys from people and patients, medical records from patients and people, and then also tissue samples and blood samples from patients and people. And we take all that into the lab, and we can do all the studies with that, Dr. Allen and Dr. Perarsky gave you a little bit of a flavor on. And then we want to take those results that Dr. Allen and Dr. Perarsky get and move it back out and study it in bigger numbers of tissue samples and blood samples from people and patients, so we can see how it looks at the population level. And then coming back to what we talked about at the beginning, we want to combine all this biological information we're getting with other information such as society level, neighborhood level, and institutional level determinants of health, things like racism, also diet, lifestyle, pollution, environment, and access to care, institutional factors, and bring all these factors together and look at the interplay between all of them. And then we're hoping that all this information will lead to new ways we can diagnose cancers, new ways we can treat cancers, and new educational strategies for cancers that we can bring back into the community and the clinic to try to address these clinical challenges that we see and the disparities, cancer disparities that we see. So this is our team. Unfortunately, 3 of them have joined post-COVID and the pandemic, so they weren't in our lab picture when we were all together. So we're looking forward to the pandemic ending so we can get a new photo taken and have everybody all together. But you definitely see Dr. Allen and Dr. Powarski here, as well as all the other members of our team. This is where we're located and what our lab looks like. So we're in a building that's off Irwin Road, sort of right down from the hospital, the Duke Hospital in Durham. So that's the building that we're in, the outside, and then some shots from the inside of our lab and lab members at work. We have joint lab space, big open lab space that we share with other labs, which is great for all working together to address cancer and different challenges with cancer. And then just like with everything in life, it takes a village to do what we do too. And so these are just a long list of not only our laboratory team, but all the people we collaborate with at Duke and outside of Duke, because it definitely takes a village to do what we do. And then we couldn't do anything that we do without funding for our work. And so these are a list of some of our funders and where our funding comes from. And I just want to end by mentioning our newest grant that we are super excited about. That's a partnership between the Duke Cancer Institute and the National Cancer Institute. And it is a grant that Dr. Nadine Barrett, Dr. Tomi Akinamiju, and I have received. And we've called our study Basic Engage. And it is a partnership between the Duke Cancer Institute and the National Cancer Institute that is focused on engaging community partners and basic scientists in collaborative research, because we really need to do a better job of working together to address the problems that we look at and all scientists look at. And we really need more engagement, I think, between community partners and basic scientists to do what we do. So we're super excited about this. We're excited to launch some cross training that we'll be doing, hopefully educating scientists in engaging with the community partners, and also in training community partners to engage and work together with scientists so we can all work together better. And we're hoping to launch some specific studies in prostate cancer and other cancers where community partners and scientists like us can work together to develop projects together and submit grants and get those projects hopefully funded. So I think you'll be hearing more from us. Hopefully that'll be a good thing. But we would love to really engage at a deeper level with everybody. So we look forward to more opportunities that come from this or from this new grant funding. So thank you so much again for having us. We'll be happy to take any questions, and happy to share email addresses and follow up afterwards as well, if that's useful.
Yes, thank you very much for the excellent presentation, how you all have brought a topic that was highly scientific and technical down to a level that people can understand. So I do have a couple of questions. It's really important that we share this type of information with community members, because we want, especially those in medically underserved populations, to be more involved in research and clinical trials. It is really important to make sure that people understand information before they get a diagnosis of cancer, because once you get a diagnosis of cancer, your world is just turned upside down. You're not thinking straight. But we like to tell people there are multiple ways on how you can participate in research. You can, like you described before, Dr. Friedman, people can give blood samples. They can give tissue samples. They can just complete a questionnaire. So there's multiple ways on how you can participate in research, and not all times will it exactly help you, but it may help the generations behind you. And so one question that I have is, a participant wanted to know, how do people participate in studies like this where they look at genes?
So Dr. Allen, I want to put you on the spot, but would you like to start and maybe take that one?
Sure, sure. So that's an excellent question. So how can you participate or get involved in studies like this? So there's I guess, so for the studies that we had mentioned, a lot of these were done with patients who were enrolled in clinical trials. And so that's the, I guess, at least for these type of studies that we had mentioned, that would be the primary way that someone could get involved. And what that would look like is, all throughout usually the year, there for specific types of either cancers or diseases, there are clinical trials that are always trying to get participants to either join or participate. And what that might look like is if you are a patient at the hospital, or even if you are just someone who meets a certain criteria for being part of a clinical study, you can talk with either your primary care physician or the hospital that you're with to ask or see if there are any clinical trials that you may be eligible to participate in, and just let them know that you're interested and that you're willing to find out more information about if you can join. And so a lot of times what will happen with these is you'll consent to share maybe some of your tissue or some of your information that researchers like us can then have access to and study more in the lab, and then help us make better or more informed decisions about how diseases or cancers are progressing. And it's really valuable to have this type of information because cancer is such a multifaceted disease. So having access to actual patient information is something that really gives us a lot of valuable information. So I would say that would be the best way to get involved if you're interested, is talk with either your hospital or your primary care physician, and see if there are any opportunities for you to participate in clinical trials or clinical studies that are going on in the area, or at Duke, or at other places that you might be seeing that.
And Doctor, I will add that, for those who do not receive their care at Duke, you can contact the Duke Cancer Center Office of Health Equity, and we can steer you in the right direction to a clinical trial that may not necessarily be at Duke, but it may be another facility. And we could walk you through the process, how to find the particular trials that you may be eligible for. Excellent. So another question I have is, how do you use DNA to determine if a person takes a DNA test? How do you use that DNA test to determine what type of cancers that person may be predisposed to?
Dr. Porowski, do you want to take a stab at this one?
Okay, so
Great.
all right, so yeah, so there are a couple methods, like for instance, there are some cancer, like there are different genes that are shown to be overexpressed in certain cancers. So for instance, BRCA gene is in breast cancer. You have highly elevated levels of that particular protein, you're more susceptible to developing breast cancer. Same thing with prostate cancer. You have prostate specific antigen, which is PSA. So there are certain protein markers that you could look at, and then there are that you could identify if they're highly expressed. Then that could be an indicator of, being able to develop cancer more aggressively or more likely. There are certain also mutations, in that occur in certain genes that actually also promote cancer progression. P53 is one of them. So that's another gene, and that P53 is most cancers, if not, all cancers. So there are certain genes that have been able to be mutated and overexpressed in a way that does promote cancer progression for certain cancer types. There are, but it all depends on what cancer and gene specificity, but there's still so much more we need to learn from that.
Another question we have in the chat is, are there language medical interpreters available to assist people that want to enroll in clinical trials and research studies where their first language is not English?
So yeah, and I think, Dr. Moore, I think you and your team can probably speak to this as well. So I think, you all, so we work with clinical research teams for all of the clinical studies that we do. So the studies where we're working with patients to either get blood samples or tissue samples from patients or surveys or, any of that information, access to the medical record for more information. Whenever we work with a study that's trying to interact with patients and get that, we work with clinical research teams that, and they at Duke, those clinical research teams definitely have people that can interpret for people that speak different languages. And so they would interact with those people and those patients to do that. And then I know, and Dr. Moore, like I said, you and your team can speak more to this, that I believe, we've also partnered with Dr. Moore and his team through community events and through getting access to people and patients through patient navigators that Dr. Moore and his team utilize to help people and patients navigate the healthcare system. And I know you all can, speak to definitely, that you all have people on your team that definitely likewise, like the clinical research team, can interpret for patients and people.
Right, and I would say generally speaking, that is an area that a lot of academic institutions need to do better in. I mean, we're not great at it, but a lot of research teams, we need to make sure that we're hiring individuals that's able to speak to individuals that their first language is not English, because oftentimes, when you see an individual, when you introduce them to a clinical trial, when you look at the time it takes to get somebody who can speak that language, you might have missed an opportunity. So that's something that I know Duke is looking forward to improving. So thank you. So that's all the time we have for questions. So I really appreciate everything that you all have presented today. I do have, let's see if I can share my slides. I just want to let you all know about our next upcoming event, which is next month. The title is Colorectal Cancer Screening and Prevention. So we have 3 speakers, Dr. Julius Waga from Duke University. He's a gastroenterologist. We have Emily Bell, which is she's from the National Colorectal Cancer Roundtable. It's going to be talking about some of the programs and interventions that they have. We have Lasanya Burnett, which is from the Office of Health Equity, talking about even though individuals may want to have screenings, they don't necessarily know how to do that. So she's going to talk about how we navigate individuals and talk about some of the programs that we have to help increase colorectal cancer screening. That's going to be done on March 22nd at 6:30. And here's the registration link. But what we'll do is, if Kirsten, if you could put that link in the chat, that would be great. And we want to thank you all for your time today. We know it's been in the evening, so we want to respect your time. But if you have any questions about any material that was presented here, or if you had any questions that was not answered because of time, please reach out to us and we'll try to get those responses to you. Here's our phone number as well as our website address. And we want to thank you again for your time, and we wish that you all have a great evening. Thank you all very much.
Absolutely. Thanks,
Thank
everybody.
you.
And thanks to
Thank
all
you,
the
everybody.
presenters.